Dear students, in this session we are going to discuss about nitrogen cycle. So, nitrogen cycle, this is a biogeochemical process through which nitrogen is converted to many forms. During these conversions, it passes from the atmosphere to the soil to the organisms and back to the atmosphere, and thereby completing a cyclical transformation. Even though nitrogen gas makes up nearly 80% of Earth's atmosphere, it is often the limiting nutrient that limits primary production in many ecosystems. This is due to the fact that the nitrogen gas in that form is not biologically available for plants and animals. These are the stages in a nitrogen cycle: nitrogen fixation, nitrification, denitrification, nitrogen assimilation, and ammonification. This is a representative image of nitrogen cycle. Now we are going to discuss about the various stages in nitrogen cycle. The first one is nitrogen fixation. So what is nitrogen fixation? It is the conversion of nitrogen to biologically available forms of nitrogen. For nitrogen to be available for use in biological systems, it must first be converted to biologically available nitrogen, and this process is known as nitrogen fixation. As we know, nitrogen gas is a very stable compound and have a triple bond between the nitrogen atoms, and therefore it requires a large amount of energy to break this bond to convert nitrogen into any other form. Since this is a highly energy required process, only a few groups of prokaryotes are able to carry out nitrogen fixation. Some levels of nitrogen is also fixed abiotically. So, there are three types of nitrogen fixation, and they are atmospheric fixation, industrial fixation, and biological fixation. Atmospheric fixation occurs spontaneously due to lightning. Only small amount of nitrogen is fixed or converted to biologically available nitrogen through this process. The second type is industrial fixation. The Haber process is an example which is used to make nitrogen fertilizers. And the third and most prominent mode of nitrogen fixation, through which 60 to 90% of biologically available nitrogen is formed, is through biological fixation. This is generally accomplished by certain bacteria and blue-green algae. So coming to the details of biological nitrogen fixation. Nitrogen fixation is carried out by both aerobic and anaerobic prokaryotes. Under aerobic conditions, a wide range of free-living bacteria such as Acetobacter, Azospirillum, etc. are capable of fixing nitrogen. Under anaerobic conditions, nitrogen fixation is carried out by free-living bacteria such as members of the genus Clostridium. Some cyanobacteria such as Ammania, Oscillatoria, etc. are also capable of nitrogen fixation. They fix nitrogen in aquatic environments. Another category of nitrogen fixers are symbiotic nitrogen fixers. These symbiotic nitrogen fixing bacteria develop symbiotic associationships with plants. Examples of such symbiotic nitrogen fixers are Rhizobium, Bradyrhizobium, etc., which form symbiotic associationship with legumes, Frangia, which associate with many woody shrubs, and Ammania, which forms symbiotic associationships with Azolla. Most of these symbiotic relations or associations are very highly specific and they have complex mechanisms to maintain the symbiosis between the participating members, between the plant and the bacteria. For example, root exudates from the legumes. They serve as signal to attract the bacteria like rhizobium to the roots, and there are a series of events through which the bacteria will be uptaken by the root, and when they reach the root they will form root nodules where they can carry out nitrogen fixation. So there are a wide variety and diversity among the nitrogen fixing microorganisms. Among all these organisms, apart from all these physiological and phylogenetic diversity among the organisms, all have a similar enzyme complex called nitrogenase that catalyzes the nitrogen fixation or the reduction of nitrogen to ammonia. The nitrogen fixation process involves a sequence of reduction reactions that require energy. So, this reductive nitrogen fixation by the nitrogenase enzyme results in the formation of ammonia, which is immediately incorporated into organic matter as amines within the microbial cells. This reductive process is extremely sensitive to the presence of oxygen and therefore strict anaerobic conditions are required for nitrogen fixation. Aerobic microorganisms which are capable of nitrogen fixation have evolved different mechanisms to protect their nitrogenase enzyme from oxygen. Some examples are physical barrier such as heterocyst present in cyanobacteria which provide a low oxygen environment for the successful functioning of the enzyme for nitrogen fixation. So, nitrogen fixation in cyanobacteria occur within the heterocyst under low oxygen tension. Some other organisms have oxygen scavenging molecules such as leg hemoglobin which is present in the root nodules of leguminous plants. So in symbiotic nitrogen fixation, the low oxygen concentration is maintained by this particular pigment leg hemoglobin which will provide oxygen concentration low enough to allow the enzyme to function, and at the same time it provides enough oxygen concentration for the bacteria for aerobic respiration. Some organisms maintain high rates of metabolic activity that keeps the oxygen concentration low enough for the functioning of nitrogenase. Some photosynthetic nitrogen fixing organisms fix nitrogen only during night when their photosystems are dormant and there are no production of oxygen. So these are various mechanisms which are evolved by different microorganisms for maintaining an anaerobic condition where the nitrogen fixing enzyme nitrogenase will function. The nitrates and ammonia which are formed during nitrogen fixation are assimilated into different organic compounds of these nitrogen fixing organisms, algae and higher plants. When animals ingest these organisms, these primary producers, then the nitrogen will reach their body. The next process in nitrogen cycle is nitrification. This is a process carried out by nitrifying bacteria which transforms soil ammonia into nitrates. These nitrates can be utilized by plants from soil. So during nitrification, ammonia is first converted to nitrite and then to nitrate. Nitrification occurs through two steps. Mostly nitrification is carried out by prokaryotes under aerobic conditions. The two distinct steps of nitrification are: the first step is the oxidation of ammonia to nitrite and the second step is the oxidation of nitrite to nitrate. The first step, the oxidation of ammonia to nitrite, is carried out by certain microorganisms which are known as ammonia oxidizers. There are two types of organisms, aerobic ammonia oxidizers or ammonia oxidizing bacteria AOB, which are present in soil, and ammonia oxidizing archaea AOA, which are present in oceans and soils. Examples for ammonia oxidizing archaea are Nitrosopumilus maritimus and Nitrososphaera viennis. And examples for ammonia oxidizing bacteria are Nitrosomonas, Nitrosospira and Nitrosococcus. These aerobic ammonia oxidizers, either archaea or bacteria, they are capable of converting ammonia to nitrite. There will be an intermediate formation, the formation of hydroxylamine. Two enzymes are required for this process, ammonia monooxygenase and hydroxylamine oxidoreductase. Since this process of oxidation of ammonia to nitrite, it releases a very small amount of energy, and therefore these ammonia oxidizing organisms are very slow growing organisms. The second step in nitrification is the oxidation of nitrite to nitrate. This is carried out by a group of bacteria known as nitrite oxidizing bacteria. Examples are Nitrospira, Nitrobacter, Nitrococcus, Nitrospinum, etc. This process also yields very small amount of energy, and as a result, these organisms are also capable of very slow growth. The complete nitrification means the complete oxidation of ammonia to nitrate. Ammonia get oxidized to nitrite and nitrite get oxidized to nitrate, and then only complete nitrification happens. The ammonia oxidizers and nitrite oxidizers, they are ubiquitous in aerobic environments such as soils, estuaries, lakes, ocean environments, etc. In waste water treatment facilities, they are very crucial since they help to remove potentially harmful levels of ammonium. And in aquaria, they help to maintain the healthy aquaria by facilitating the removal of toxic ammonium excreted through fish urine. Next is anaerobic ammonia oxidation, which is also known as anamox. This is carried out by prokaryotes belonging to the Plactomycetes phylum of bacteria. An example of a bacteria capable of carrying out anamox reaction is Brocadia anamoxidans. They oxidize ammonia by using nitrite as the electron acceptor. They carry out this in the absence of oxygen. Anamox bacteria are found in low oxygen zones of ocean, coastal and estuarine sediments, mangroves and freshwater lakes. This process is responsible for a significant loss of nitrogen in ocean. The next process in nitrogen cycle is denitrification and nitrate reduction. Denitrification is a process that converts nitrate to nitrogen gas. So after denitrification, the nitrogen gets escaped from soil back into the atmosphere. So this process removes bioavailable nitrogen from the ecosystem. Even though nitrogen gas is the ultimate product of denitrification, some other intermediate gaseous forms of nitrogen are also formed, such as nitrous oxide, nitrite, etc. Nitrous oxide is a greenhouse gas and nitrite is involved in the formation of certain carcinogenic nitrosamines. As we said, denitrification reduces or removes bioavailable nitrogen. It contributes to greenhouse gas accumulation, and also it may result in the formation of carcinogenic nitrosamines. This denitrification is an anaerobic process. It occurs mostly in soils and sediments and also in anoxic zones in lakes and oceans. Examples of some denitrifying bacteria are Bacillus, Paracoccus, Pseudomonas, etc. Pseudomonas denitrificans is a denitrifying bacteria. Denitrification is highly important in agriculture because this process results in the loss of soil fertility. Denitrification in wastewater treatment is very beneficial since it helps to remove unwanted nitrates from wastewater effluent. Nitrate reduction. This is the process by which nitrate ions are reduced. There are two types of nitrate reduction: assimilatory nitrate reduction and dissimilatory nitrate reduction. Assimilatory nitrate reduction is done by a heterogeneous group of microorganisms including bacteria, fungi and algal species. During this process, the nitrogen ions are assimilated or incorporated into the organic matter in the cells. This process involves several enzyme systems including nitrate reductases, nitrite reductases, etc. And during this process, ammonia will be formed, which is incorporated into amino acids. So this is a process of assimilation of nitrate. The second type of nitrate reduction is dissimilatory nitrate reduction. This is also known as nitrate respiration, and this occurs in the absence of oxygen. Here, nitrate is converted to a variety of reduced products, and for their reduction, organic matter will undergo oxidation. There are two types of dissimilatory nitrate reduction. In the first type, facultatively anaerobic bacteria such as alkaligens, Escherichia, Bacillus, Nocardia, etc. reduce nitrate to nitrite under anaerobic conditions. And these nitrites are generally excreted, or sometimes they may get further reduced to ammonium. Since there is no nitrogen gas here, no denitrification occurs. In the second type of dissimilatory nitrate reduction, for example, by Paracoccus denitrificans, Thiobacillus denitrificans, etc., the nitrate is converted to nitrite, then to nitric oxide, to nitrous oxide, and then finally to nitrogen. So here nitrogen gas is formed and denitrification occurs, that nitrogen gets released into the atmosphere. So nitrate reduction, there are two types, assimilatory and dissimilatory nitrate reduction. And in dissimilatory nitrate reduction, again there are two types. First type, nitrate to nitrite and sometimes to ammonia. And then in the second type of dissimilatory nitrate reduction, the nitrate is converted to nitrogen. Next process in nitrogen cycle is nitrogen assimilation. This occurs when inorganic nitrogen is used as a nutrient and get incorporated into biomass. Ammonium ion can be directly incorporated without any major energy cost, but when nitrate is assimilated, high energy requirements are there. The nitrate must be reduced with a significant energy expenditure. And during this process, sometimes nitrite may accumulate as a transient intermediate. So nitrogen assimilation is a process through which inorganic nitrogen get incorporated into biomass. The next process in nitrogen cycle is ammonification. Nitrogen in the tissues of organisms. It is present in the form of organic nitrogen, and it gets decomposed when the organism excrete waste or when the organism dies. So this organic material will be decomposed, these tissues will be decomposed by various fungi and prokaryotes, and they release the inorganic nitrogen from these tissues back into the ecosystem as ammonia, and this process is known as ammonification. The ammonia then become available for the uptake by plants and other microorganisms for their growth. So, we were discussing about nitrogen cycle. There are several processes involved in nitrogen cycle such as nitrification, nitrogen fixation, denitrification, nitrate reduction, nitrogen assimilation and ammonification. Thank you so much for listening.